Okay, we are recording now. So this video is about review of eye anatomy. So we're going to talk about the layers of the eye. Another way to talk about the layers is to call them tunics. So really in anatomy, anytime anything has multiple layers as part of its overall structure, we call them tunics. So that's not limited to the digestive system or the arteries and veins. You might be thinking like, oh, tunica media, et cetera. A tunic is just a layer of an organ or a structure. So let's do the outermost first. And this is going to be, actually, you know what? I have a better idea. If I use the circle tool, then it will actually be round without me having to try and draw a circle. And then I'm going to do this. So what I've just drawn is the sclera. And then what I'm going to draw now is the cornea. And the reason I erased that little spot is because the cornea is actually its own sort of dome. So it goes a little something like this. And I'm going to try and draw that again because that's a lumpy cornea. That's probably what my corneas look like since I have astigmatism. All right. So let's just clean up that edge a little bit there. So both of these layers are made out of fibrous connective tissue. It's just that the sclera is not transparent. The cornea is transparent. So this is called the fibrous tunic of the eye. It's also sometimes called the uvea, which means grape. It's the connective tissue structure that gives your eyeball its shape. Actually, because I need to draw the palpebrae, I'm going to relabel it like this. So I just mentioned the word palpebrae, which is plural. Palpebra is singular. And that is a fancy science word for your eyelid. And then lining the inside of the eyelid and the surface of the eyeball is a layer called the conjunctiva. And it's not really green, but I'm going to draw it as being green so we can see it. So it lines the inner surface of your eyelid, and then it flips around and lines the surface of your eyeball up until the cornea. It does not cover over the cornea. So same deal here. So this is why you can't get a contact lens lost behind your eyeball. There's a blind end right here that prevents that. So it's not the case that you would be able to stick something behind your eyeball successfully. Your conjunctiva prevents it. The reason the conjunctiva does not cover over the surface of your cornea is because your conjunctivae, plural, have blood vessels in them. So if you had your conjunctivae covering your corneas, you would have this constant overlay of blood vessels in your field of vision, and that's not ideal. And this one is So conjunctivitis or pink eye is an infection of that layer specifically. And unfortunately and disgustingly, the most common route of transmission for pink eye is the fecal eyeball route. So you end up touching some poop somehow and then rubbing your eye, and then you get pink eye. This is why it's more common in little kids. Okay, so that's the outer structures of the eye. So we have the conjunctiva not covering over the cornea, and that's because the cornea is transparent and light goes through there so you can form an image, which means that you need it to be free and clear of any obstructions. But the cornea is still made of living cells. And what that means is it can't have blood vessels in it because you would see them, but it still needs oxygen and nutrients. And so there's inner structures of the eye that help with that. So I'll explain what that little blue dot is momentarily. So now we have this smooth muscle structure that is the iris. And then on the anterior surface of the iris, there's melanocytes that make pigment. And that covers over the smooth muscle and also gives you your eye color. And then behind that, we have an additional smooth muscle structure. And I'm going to color that in. And this is the ciliary body. And I'm drawing a sagittal section of an eye, and that's making it appear as though it's at the top and at the bottom, but really it goes all the way around. So the ciliary body is a circular structure of smooth muscle. And one of its jobs is to support the lens, which looks like this. The lens is also transparent, but it is made of living tissue. So again, we have this problem where we have a bunch of structures that need to be transparent because we don't want to be trying to form an image while we're, geez, I'm trying to silence my phone and it's not cooperating. Shush, there we go. We have a bunch of transparent structures and we can't have blood vessels in them because then we'd see those blood vessels. So luckily, we have the ciliary body because in addition to supporting and holding the lens via suspensory ligaments, it also contains choroid plexuses, which I'm going to kind of, it's not dark enough. Let's try another color here. So also in the ciliary body, in addition to the smooth muscle, are ciliary processes. And these are just sort of little finger-like projections of the ciliary body, and they contain a choroid plexus. A choroid plexus is just a specialized capillary bed that is designed to have something filter out of it. So in this case, it's aqueous humor of the eyeball. So the aqueous humor comes out of the ciliary processes, and it bathes the lens in oxygen and nutrient-rich fluid so that those lens cells stay healthy. And then it flows out of your pupil and kind of swirls around. And that helps to bathe your cornea in nutrients and oxygen as well to keep those corneal cells healthy. And then it eventually flows into this little dot that I've drawn right here. The special name for the spot where the cornea transitions into the sclera, so where it goes from being clear to being white, that's called the limbus. And at the limbus, there is a structure called the scleral venous sinus, also known as the canal of Schlemm. So let's and that's the venous structure that takes up the used aqueous humor and puts it back into venous circulation, so it recycles it. So ideally, what should be happening is that your choroid plexus in your ciliary process should be producing aqueous humor at the same rate as the aqueous humor is being absorbed, so that intraocular pressure stays stable. When you go to the eye doctor, they actually test your intraocular pressure. I've been to the eye doctor recently, so this happened to me not that long ago. They have you put your chin on a little chin rest and then open your eye and stare at, typically it's like a picture of a hot air balloon, and keep your eye open, and then they aim a little poof of air at your eye and take a picture while that's happening. So what they're doing when they do that is poking your eye with the air to see how much deflection that produces. And that's a way to measure intraocular pressure without having to poke you in the eye manually. So although the puff of air is uncomfortable, it's far preferable to being jabbed in the eye by your ophthalmologist. So aqueous humor recycling is responsible for intraocular pressure. All right, so that's the front stuff. So let's continue labeling. So we've got the ciliary body, and I'm going to erase my title because I need some more room here. Ciliary body consists of muscle, ciliary muscle, which is smooth muscle, and ciliary processes, which are the little finger-like projections that produce aqueous humor. And then these little guys that are holding the lens in place have two names. One is suspensory ligaments. The other is a fun one. It is ciliary zonule, which is a fun, jazzy sounding word. So I usually call them ciliary zonules because it's just a more fun word to say, but suspensory ligament is fine. So the way that these help you focus an image is the smooth muscle of the ciliary body contracts or relaxes, and that's going to pull your lens into a more flattened or more rounded shape. And that's going to help focus light from either a near or a far object on the back of your retina in the appropriate spot. So as we all age, the connective tissue of all of our body, really, so as you age, your cells get worse at making high quality collagen, which means that your connective tissues become more brittle and stiffer. Same deal is true with the cornea and the lens. So as we age, the ability of our lens to flex while focusing is not as good as it used to be. And that means that we end up having a hard time going from focusing on far objects to focusing on near objects. So that gradual stiffening of the lens is why most people end up needing reading glasses as they age. It's a normal part of aging. Okay, so now we're in behind the lens territory. So this is the posterior chamber of the eyeball. And covering over the back of the ciliary body and just going in general between the sclera and the retina is this heavily pigmented layer called the choroid. It's also called the vascular tunic of the eye. And I'm making it brown because it's got a lot of melanin in it. So if you go and look at it under a microscope, it looks brownish black and it has a lot of veins and arteries in it. So let's draw some of those. All right, so it's heavily pigmented because we don't want stray photons to go through our retina if they're missed by the photoreceptors and then go hit something they're not supposed to. Photons are high energy particles, and your eye is specially designed to be good at catching photons. But if they go to the wrong place, they can cause problems. So we have this elaborate pigment system to kind of try and catch stray photons in case they don't manage to hit one of your rods or cones. So let's label that layer. Okay, and now I need to do some erasing because I need to create the optic disc. So now we're into the neural layer of the eyeball, which is the retina. And this is the layer that's actually capable of producing a visual signal. So it's a specialized array of cells that are good at catching light and turning that light into a receptor potential, which is then turned into an action potential via your optic nerve. And so your retina is 3 cell layers thick. And we'll have a little zoom in on what those cells look like in a minute, but and then the axons of these cells that are collecting the visual signal, they have to escape the eyeball somewhere. So they do so at a place called the optic disc, which is where all of the axons pass through the choroid and through the sclera. And this is also your blind spots. So you don't have any photoreceptors here, it's all axons. So optic disc / blind spot. And then additionally, if we go through the center of the lens, so straight through the visual axis, all the way back. So let's draw the visual axis here. So if you go straight back from the lens through the center of the pupil, you end up on this little dent in the retina. And that is an area that is really, really high in cone cells, which are the cells that detect color. And it's relatively low in rod cells. And it's also the densest area of photoreceptors. So it's your area of highest visual acuity. And that is called your Fovea Centralis. So the Fovea Centralis is the name for the divot. It sits in an area of the retina that is lower in blood vessels called the macula. And macular degeneration, which is another disease of the eye that's associated with aging, is a fairly prevalent cause of blindness in older individuals, and that refers specifically to atrophy of the macular area of the retina, which includes the fovea centralis. Okay, so that is a sagittal view of the eye, including all of its tunics and all of its necessary parts. So now let's zoom in on the retina itself. So I'm going to delete this picture and we're going to draw a more detailed picture of the retina. So to give me a starting point, I'm going to draw myself some sclera. And then I'm going to draw myself some choroid. Let's put some blood vessels in there. And now we can start drawing our visual layer. So how do I want to, I think we'll do. So rod cells look like this. Actually, let's make that thinner. So they're a really weird shaped cell. They're almost shaped like a pocket comb. And these are the cells that detect light or dark. So they're for edge detection. They're good for making out silhouettes and patterns. They do not detect color. That is the domain of cone cells, which are shaped similarly, but they're more triangular. And those are for your color vision. So I'm not going to draw them in this much detail in my little slice of retina over here, because that would take approximately 80 million years. So I'm just going to draw them like that, or like this. So before we do that, I need to draw the pigmented epithelium. There's a small epithelial layer that is just right here. And the tips of the rods and cones stick into this layer. Let's draw some rods. They are more numerous than cones in the majority of the retina, and because they're straight, you can pack more of them in. Okay, so the photoreceptive portions of these cells are arranged like that. And then we have a layer of, and I'll label all these layers in a little bit, bipolar cells, which are very short neurons. They are bipolar neurons. And their dendrites have lots of processes. So they have a single dendrite with lots of extensions. And they're receiving input from multiple rod or cone cells. So you have this decrease in cell number. That's a significant decrease as well. So there's many, many rods and cones. There are fewer bipolar cells. And then there are even fewer ganglion cells. Which receive input from multiple bipolar cells. And these are the cells whose axons travel the surface of the retina and out through the optic disc. And then ganglion cell axons form the optic nerve. So one thing it's important to mention is that the path of light through the eye means that a photon, which I'm going to illustrate here as being a little orange guy, a photon has to travel through all of the axons, past the ganglion cells, past the bipolar cells, and then hopefully it lands in one of the photoreceptive parts of either a rod or a cone. And that creates a receptor potential that gets transmitted through the retina. So the path of light is this way. The path of the neural signal is rod and cone excites a bipolar cell excites a ganglion cell. So the path of a photon and the path of the electrical signal that it produces by landing on a rod or a cone, they're in opposite directions. And this is why we have a blind spot, because you have to have somewhere for that information to go and exit the eyeball. And because our retina is built this way, there's no way to do that without making a little spot where the axons leave and where there's no photoreceptors in place. Octopi and cuttlefish and squid have roughly the same eye as we do. So it has all the same parts, except for their retinal layers are flipped. So their rods and cones are out front, their bipolar cells are behind that, and their ganglion cells are behind that. So they don't have a blind spot because their retina is identical to, but opposite from ours. So that's evidence that the camera eye, which is the kind of eye we have, has evolved several times. And there are creatures who evolved a slightly more efficient camera eye than we did, which is interesting, but everything else is pretty much the same. Okay, so in order to form an image, and let's draw a mini eye over here. We've got so the iris is going to control how much light gets to enter the eye. So your parasympathetic nervous system tries to keep your pupil small so that you're not getting blasted with light all the time. And then your sympathetic nervous system is in control of the muscle that causes dilation. So they're kind of constantly pushing against and pulling on each other to regulate your pupillary diameter according to how much ambient light there is. So a bunch of light is always aimed at your eyeball, but only some gets through. And due to the thickness change of going from a fluid aqueous medium to this now dense collagen, your lens actually focuses the light, but also flips the image upside down. And it focuses the upside down image on your fovea centralis. And then via the optic nerve, let's move this down here, via the optic nerve, whatever excitation that results forms an image that gets sent to visual cortex. And visual cortex is on your occipital lobe of your brain, remember, and it's actually the visual cortex that flips the image back right side up. So your image that you're catching on your eyeball surface is inverted because of the way that the light is refracted through your cornea and through your lens. And then your brain is like, don't worry, I got this. We'll just flip it back around in post-processing. We're all good. So that is a review of the anatomy of the eye and also how that relates to the eye's function.